This is Sarah Wilkinson from Humber College and the University of Guelph-Humber. In this video, I'm going to continue my series on ATP generating pathways, and we're going to start to talk about the ones that use oxygen. So before we can do that, we have to talk about the formation of acetyl-CoA. And acetyl-CoA can be formed from many different substrates. In this video, I'm going to talk specifically about its formation from carbohydrate and fatty acids. In previous videos, I've covered enzymes in chemical reactions, what ATP is, several other ATP generating pathways included the phosphagen system and anaerobic glycolysis, and a little bit about coenzymes, so NAD and FAD. I would encourage you to watch those before you watch this one. In this video, like I said, I'm going to be talking about the formation of acetyl-CoA. Just a disclaimer, my molecular drawings are not necessarily accurate. They're very simplified for explanation purposes. So if you're taking a biochemistry class, this is not sufficient for your biochemical knowledge. It's just a general concept. There are really three essential steps for using oxygen to produce ATP, so for oxidative metabolism. The first is the formation of acetyl-CoA, which we're going to talk about in this video. The second is the oxidation of this acetyl-CoA in the Krebs cycle, where the coenzymes are going to be reduced, so adding electrons and hydrogens. And the final step is the formation of ATP in the electron transport chain, where all those coenzymes that have been accumulated in multiple ATP generating pathways are going to be oxidized. This video is going to cover the first step, the formation of acetyl-CoA. So let's first start talking about carbohydrates, how acetyl-CoA is generated from carbohydrates. If you recall from anaerobic glycolysis, 2 3-carbon pyruvate molecules are formed in the cytosol. And just to orient you to this diagram here, this is the outer membrane of the mitochondria. This is the inner membrane of the mitochondria. And here is the inside of the mitochondria, and we call this the mitochondrial matrix. The pyruvate formed during anaerobic glycolysis can move into the mitochondria. I'm not going to go into the details of how this happens, just know that it can happen. So this 3-carbon molecule moves into the mitochondrial matrix. Before it moves on to the Krebs cycle, it has to become a 2-carbon acetyl-CoA molecule. Acetyl-CoA is essentially 2 carbons, and there's other things attached to it. So remember, this is very simplified. And CoA, again, is a coenzyme. So how does this happen? Well, one of the carbons is lost as carbon dioxide. And if you remember, part of metabolism, we're producing carbon dioxide. So this is one of the steps that produces CO2. So the 3-carbon molecules becomes 2, 1 carbon is lost through carbon dioxide, which can then leave the mitochondria, leave the cell, and into the bloodstream so that it returns back to the lungs. Another step within this reaction is the reduction of the coenzyme NAD. So electrons and their associated hydrogens are removed from the molecule of pyruvate, so energy is transferred to NAD in the form of NADH, which can then move on to the electron transfer chain. Finally, because this is acetyl-CoA, a molecule of CoA is attached to acetyl-CoA. This whole reaction can be written out as pyruvate plus NAD plus CoA, becoming acetyl-CoA, carbon dioxide, and NADH. And because every reaction needs an enzyme, here's the enzyme for this one. So this one's called Pyruvate Dehydrogenase. Pyruvate Dehydrogenase. That's how you can remember that. It's removing hydrogens, dehydrogenase. Fatty acids become acetyl-CoA in a little bit of a different way. If you remember, fatty acids can be broken down from intramuscular triglycerides in the cell, or they can come into the cell from the blood. The fatty acid will then be bound to a molecule of CoA to become fatty acyl-CoA. This can then pass into mitochondria. The way fatty acids are metabolized is through a series of reactions called beta oxidation. All fatty acids are even numbered. So 2, 4, 6, 8, 10, 12 carbon molecules. And if you remember what acetyl-CoA is, a 2-carbon molecule. What this beta oxidation will do is will cleave 2 carbons at a time. So every round of beta oxidation, okay, produces 1 molecule of acetyl-CoA. In the process of forming acetyl-CoA, the fatty acyl-CoA becomes 2 carbons shorter. So if we start with an 18-carbon fatty acyl-CoA, 1 round produces acetyl-CoA and a 16-carbon molecule. Next round, another molecule of acetyl-CoA, then 14 carbons, and so on and so forth. And you can see how you end up producing quite a bit of ATP when you have fatty acids as substrate because they're so long. In each round, NAD and FAD becomes reduced. So electrons and hydrogens are removed from the fatty acyl-CoA, transferred to NAD, and again, this NADH and FADH2 can then go on to the electron transport chain. Just to summarize where acetyl-CoA comes from, when anaerobic glycolysis generates pyruvate, pyruvate can then move into the mitochondria, carbon dioxide is removed, a few other things happen, and you end up with a molecule of acetyl-CoA. When fatty acids are involved, they undergo beta oxidation in the mitochondria and acetyl-CoA is produced. Didn't talk specifically about protein and amino acids because our body prefers not to use them as substrate, but if it does, all the different amino acids can enter at different points of the Krebs cycle or acetyl-CoA because remember there's lots of different amino acids. I'm not going to go into detail in this video, but proteins and amino acids enter, depending on which one there is, somewhere in here. The next video will go on to show how acetyl-CoA enters the Krebs cycle and is oxidized to further generate FADH2 and NADH. So as you can recall, there are 3 essential steps to oxidative metabolism. In this video, we covered the formation of acetyl-CoA from carbohydrate and fatty acids. We're going to go on to the next video to show how the oxidization of acetyl-CoA in the Krebs cycle further reduces coenzymes, and finally we'll finish talking about the electron transport chain.